the nuded bamboo coral colony. It's on
Okay,
the right.
see that?
Yeah,
Yeah.
and there's a little white
move initiated.
peg
Move initiated. Thanks, Rich.
to the right of it.
Okay.
Just wondering if that's something.
It looks like a white dot.
Yeah, I see it.
Sorry, it's. I know it's a sort of weird, but sometimes I
Coming
get,
up in the middle of the lasers now, or right about now, that tiny little thing.
Yeah.
Okay.
Sometimes the best things are those funny little blips that my eye catches.
All right, let's go in there and see if I can.
And then sometimes they're just dead stalks, aren't they?
Yeah, I know.
I could probably do a snap from here.
Okay, yeah, go ahead.
Oh, but
It's
not
a
this
snail.
time.
It's a snail.
Okay, you saw something.
Oh, that's the first. Oh, good. So, you know. Oh, and there was a. Oh, it's a shrimp. Okay. I thought that was. Yeah. Okay. Yeah. So let's just briefly just take a look at the snail.
Okay.
You have
I can come back
another
out if you
little
want.
Yeah, please.
cauterizing sponge.
Let me get
front
in
of
there.
that
I
coral
can.
too.
Yep, we're full wide.
Yeah. Okay. Thank you.
What's that? Yeah.
What's that, Bob? Oh. Yeah. There's a. Looks like there's a little field of. That little patch has just maybe another. Is it another snail? Oh, it's fine.
This is a large whipped bamboo coral over to the left. The Lepidopsis group is what we are calling them until they get their new names or monikers.
See, this is what's interesting to me. You know, I mean, is it? Maybe it's the snail that's been causing it, but this bamboo coral is. It's like all the tissue from the bottom up seems to be removed or gone.
Okay, zoom in.
All right.
It's up and down.
Yeah, some snail that I don't recognize, but very pretty. Looks like it's got some very nice ornate whorls on the shell. For all I know, it's what was eating that coral. Yeah, that's great. We can move on.
Okay.
Yeah, Les was Les Watling at the University of Hawaii Biology Department.
Okay,
He was noticing that the water has gotten slightly colder here, even though we're upslope a little ways, and he was pondering whether or not this could be because the water flow is being deflected from deeper, this little micro upwelling type of thing.
Oh, interesting.
Yeah, that is a very good observation, Les. That may be what we're seeing here.
Okay, so cliff. This is good. Cliffs are good.
Cliffs are good.
All these rocky. Was it a promontory or a cliff or a?
Ledges where you can get, these little areas.
It's like a mushroom coral right at
All sorts of-
lasers.
Oh, is that an anthomastus?
By the lasers.
Sort of a red.
Where is it?
Red.
Oh, yeah.
Yeah, I see
So
it.
to the
Yeah.
left.
He's got it, I think, right there
Yeah,
under
okay.
the. You're right.
Woohoo!
That's something we haven't recorded today, I believe.
Things are a-changing.
Okay, go ahead and zoom.
So that's a partial.
Okay.
So this is indeed a mushroom coral.
Let's see if I know if I can turn on here or not. Let me get a little bit.
Pseudanthomastus, maybe? Anthomastus? Boy, I just...
Oops.
I've got to look back through my notes. What Tina was trying to...
Is that one of the polyps closed?
Where's that?
That red swelling on the surface?
Well, I don't know what that is.
That's weird.
It is weird. So
So for those of you who might not have seen one of these before, this is a mushroom coral. It's got a number of polyps that come off of this sort of mushroom-shaped central body. It's a colony, and each of these polyps has a mouth surrounded by 8 feeding tentacles. You can sort of see the channel moving from the mouth to that central mushroom, if you will. We can move on. Thank you.
Okay.
All right.
When you're clear.
ROV nav bridge, move complete.
Okay.
Move complete. Thanks, Bridge.
Okay, thank you.
There's that Chrysogorgia that was just to the left of it. If we could just do a quick zoom on that, I'm just wanted to make sure that's a brittle star.
Okay, go ahead and snap on that. Right there where the lasers are, you're talking about? Yeah, okay.
The squat lobster.
Oh, it's a squat lobster. Okay.
All right.
Again, I live in hope.
So there was a Chrysogorgia upslope a little bit.
Right,
And I'll keep an eye out for one of these, but I thought I saw a Metallogorgia, which I don't think we've actually documented today, and I'll see if I can.
Okay, we got to go off to the right here, down.
Okay.
I think it's in the direction we're heading.
All right, let me go up and to the right.
That's good.
Yeah, that move just completed. We got a couple minutes to swing. That move was at a 320.
Copy that.
I came left to keep you in view.
Oh, I got you.
So I'll
Okay,
lead
roger
you-
that. All right. Keep.
Lead you over here in a minute.
Some more of these bamboo corals. There was a little black coral to the lower left. And yeah, another one that could have been Trisopathes. Yeah, let's see.
Something in particular?
Pilot, if we get a chance, right going off left screen center, is that kind of a little brownish colony there?
The one at the top there.
If we could get a quick look at that.
Maybe. We can get a look at it. I don't know if it's quick.
Yeah, okay, well, I look at it. Where's it? Oh, there's a fish?
Oh, yeah.
Where is it?
Oh, yeah, there is
a
up
very convincing crack in the rock.
Yeah. Okay, is this the guy here you were looking at it right?
Yeah, the brown
just below
one.
the lasers.
That's right.
Okay, all right.
Not too much vert.
Yeah,
Yeah,
oops.
you just-
Okay, yeah.
That's a rock.
It's a rock.
A little bit of vert and a little bit of axial at the same time. Oh, keep
We
your
do that a
toes
lot
in
here.
there.
We identify rockfish.
Yeah.
So that
Okay, you
little
want to zoom in?
colony of black corals,
tilt.
50
Got it.
centimeters or so.
It's good practice.
That's a
Stretch
partial.
those fingers.
All right.
Partial standby.
So this guy.
Okay, a little more,
is-
Annie, please.
I don't know if Scott's still on the teleconference here. But if he is, that would help. I think Tina's probably asleep right now, even though she's got the event log on. I'm trying to look this up in the Okeanos Animal Guide.
I think that move is pretty well settled out. I think you guys are ready for another.
Maybe Trisopathes.
Yeah, you can get it going here.
Do you want to
Yeah,
do
I
320
You want to go
or?
back wide, please?
Yep.
Thank you.
So that's a tough go, Don.
Perfect.
So
Yeah.
just
Thank
one
you,
toe
Tyler.
down.
That
Yeah,
was
I
good
know.
enough.
Okay.
So it's just
Copy that.
balance axial inverts and
Yeah, as soon as I reach out to move the camera, then I let go of forward way, and the current just drug me back.
We still got a good shot, though.
I'll probably
Okay.
call that move in at 310.
310, copy.
Do you guys?
I'm
think-
All
looking
right,
at
watch lead, we can go ahead.
3-0.
Percent yes.
Pilot.
Yes, okay, thank you.
So
coming
to
right.
give you an idea of what we're
Sounds
doing,
good.
we've
I'll call it in.
today we've been
Bridge,
coming
are we
up
now?
sort of on the southern side of this ridge crest.
This is the bridge. Go ahead.
Then we
Yeah,
got
like a
to the
move,
steeper
range
part, and we're kinda
30
angling over
meters,
more toward
bearing
the
310
center and
degrees, speed 0
part
decimal 2
of
knots.
the crest as we go up slope, sort of
Range
tangentially.
30 meters, bearing
at this
310
point,
degrees.
and just kind of
speed
getting an
0
idea of
decimal
the,
two.
just
Good
what's
copy.
happening
Coming up.
on this ridge.
Okay.
ROV nav, move initiated.
Move initiated.
And maybe it's a mirage, but I think the number of corals has picked up. What do you think, Chris?
No, I think you're right. Actually, I just think it's. Well, you mean abundance or diversity?
Just abundance and
Yeah,
diversity.
abundance has-
The bamboos are coming back in
Yeah.
that we saw earlier. We're not seeing any particularly new ones that we didn't see
Yeah.
in the beginning, but we lost some of these branching ones for a little
Yeah.
while, and
Yeah.
they're coming back. If you, as you can see, you can see up slope, we've got a kind of nice little stand of stuff that continues to increase. So there's a interesting phenomenon there. If we had a moment to the lower right, pilot.
Okay, the sponge.
Yeah, and so what's going on here is the tall sponge has died, and another species of sponge has rudely decided to use it as a prop. Now he's on the other face, and I think it's probably Poliopogon, whereas this is one of these turbocharger sponges. And Matt Dornbeck coined this term. We were talking about this at lunch a little bit. It's kind of like in reference to the holes on the top might actually accelerate water current through the sponge, and I guess it's sort of like a turbocharger, and in fact it's functioning to
Interesting.
make the water flow more
Okay, Annie, you
efficient.
want to zoom in?
Yep. It's a
That's partial.
Poliopogon sponge.
Okay.
It's one sponge living
So
on another one. And there's a shrimp there too, as
Go in
well as
further
a
now.
weird
I think
Galathian.
I'm stable.
And this is always a good example of how even the dead bodies of some of these organisms provide homes for a lot of the animals in this habitat. Not only you're seeing some crustaceans, the shrimp and the squat lobster, as well as this other sponge, but you're seeing hydroids, and undoubtedly there are innumerable other tiny microorganisms living on the body of this sponge in the channels and throughout the surface. But the crustaceans use the height of the colony to take advantage of water currents to fish for food. The hydroids are doing the same thing. And space is always at a premium. Good space, I should say.
Okay, video, go wide.
Yeah, thank you, pilot, for that.
Welcome.
It's kind of an interesting little observation. Another Walteria sponge to the left there. They're pretty common today. couple dead ones to the right, so you can see what happens to sponges when they die. And a little chrysogorgid, little unbranched primnoid, possibly Norella. And got some more bamboos in the background. But another corallium, you see that one standing up that we just got an image of a little while ago.
Yeah.
Couple white things on the seafloor, but I don't think they're your sea stars.
No,
though.
they're bases of dead corals, almost certainly. Or sponges, anyway.
Yeah. Some of these primnoids are Candidella, I think. Candidella gigantea, like the one right in front of us. You're able to zoom in on one and confirm that one. That species is present here. I think the community's picking up. See, I can see a fan over there in the background there.
Yeah.
I think as we get closer to the northern side and in the steeper terrain, we're getting
ROV
more
nav,
things.
move complete.
Move complete. Thanks, Bridge.
Can we do a snap zoom on this right side, on the right side, on the rock with all the kind of, I think there's just a bench of bases, but there's some white things.
Okay, right there, you're talking about lasers. Okay, go ahead, Annie.
All right. Well, that's a partial.
Okay.
Yeah, it looks like mostly bases, but yeah, bases. Okay, we can move on. Thank you.
All right.
All right. And we're full wide.
Okay, thank you.
I keep thinking I'm seeing Metallogorgia. I know there's a little discussion, and Les was saying it's conspicuously absent here. It's a very, very common chrysogorgid coral elsewhere.
Sure looks like it, though. It's got that
I think
metallic,
I'm sort of seeing it, and then as we get closer to one of the possible colonies, it turns out it's not it.
Oh, well.
Yeah.
But it's fuzzy and pink.
There's a nice bamboo fan. I think
The Metallogorgias are kind of like
Les
a
and
flat
Scott were going,
inner plate
if I'm
on
not
top of a
mistaken,
stalk.
J-clade for this.
I
Possibly.
think so.
I got a couple minutes left to swing. How does 300 sound for the next move? 3000.
That's
I
okay.
like the terrain
that
better
works.
here. It's
Yeah.
something about it that's
That'll
very
ride this
appealing.
ridge nicely.
It's kind of a three-dimensional, I mean, it's got like cliffs and
Yeah,
call it
shadowing and
shadowing,
sediment.
texture. Yeah.
Bridge, RV now.
The bridge, go ahead.
Yeah, I'd like a move range 30 meters, bearing 300 degrees, speed 0.2 knots.
Range 30 meters, bearing 300, speed 0.2.
Good copy.
Coming up.
I am surprised with all these bamboos that we haven't seen any Hipposteria today. That just seems odd
You
to
know,
me.
I'm getting the impression that there's just something about this environment that the water, or I mean the water current, I mean maybe the water current prevents things like larvae from settling, or maybe it physically just makes it difficult for them to feed. I don't know if I've ever noticed the water current in areas where Hipposteria abundance is high, but it could be that, you know, we've watched them adhered to the surface of these corals with their tube feet. Maybe that's a thing that requires
ROV nav, move
their
initiated.
balance.
Move initiated. Thanks, Bridge.
What was that heading?
300.
300.
He's at 310 right now.
Copy that.
Coming left.
I think that'll take you nicely uphill. Kind of stay on this
Just getting caught up in the event
zone here.
event log. So Bruce is noting that we haven't seen a whole lot of fish here on this dive, and I would agree. I think we've seen only one synaptobranchid eel and maybe two grenadiers. But yeah, the oxygen doesn't seem that low here. It's over 2. It's not as bad as some places. Tim Shank is noting that he thinks he's seeing Metallogorgia as well too, and then we get a little bit fooled as we get close to him. Turns out it's another one of these stock chrysogorgids that we imaged a few times today. Tim was asking whether we've seen many
about the community that we've entered since we began today is that every once in a while you find a really abundant little bunch of those little candelabra sponges. Those, I don't know what it is about them. They're never big, but when they do appear, they seem to be in big clusters, as opposed to the ones that I think Mbari saw were just huge, right?
Yeah, they're much bigger, and even the one in the Marianas was bigger than the ones we've seen today. You know, we were easily able to grab 2 samples of that off of the Marianas, and I mean, we dealt with the specimen in our lab, and it was probably this wide, you know, about 30 centimeters wide, but I don't know how tall, easy to grab.
But these have all been really tiny things.
Yeah. Look at that sponge. Is that a cool sponge or what? Yeah, thing. It's an alien.
It is cool.
Yeah, it's neat.
Yeah.
You know what
we could. It needs a snap zoom on it. I mean, it's so cool. Again, we've imaged these already today. But
what are this? Is this Freya?
Maybe. I think so.
What, did I get it right? Actually,
no, you did get it. I'm pretty sure it is, but we got a piece of it today already. I think it is, so we'll find out later.
Okay,
so
go ahead.
What's really interesting about this is we had commensal black corals growing out of the one we took. I'm not seeing it on this one. But and then whip coral off to the left, if anyone's interested, is Candidella, another primnoid. But we're looking at this.
This one doesn't have it.
Yeah, and I actually think this could be the sponge. It's a little different morphology, but at least related to the one we collected earlier in the cruise that did not have the black coral also on it. Well, look at this. Look at this. There's some thready stuff up to the upper left.
Co-pilot, take the camera, please.
Okay.
Thanks.
I have it.
Yeah.
Right
and
there.
Just go up a little bit.
Is that it? That could be the black coral.
Is that all we got on Zoom?
Yep, that's it all.
Okay. I can get us closer if you want to.
On the other side or on this side, you mean?
On this side. Oh. You want to get on the other side of it? I can see.
Yeah, Matt Dornbach was trying to point something off, something out to me. What did you see, Matt? Let's see.
There's some kind of small feathery extrusion on the right side.
Right. So, oh yeah, right on the edge.
Yeah.
So I do think this is the sponge we collected a few days ago, or a few cruises, a few dives ago. Dive 5 is okay. Matt actually was helping a bit with the specimen processing, and he remembers that it was dive 5 when we saw
go down.
something like this. That could be the same thing.
Go down the base, man.
Yeah, look at this. It's beautiful texture, and I just don't remember all this channelization in some of the more commonly occurring farreid species. So I think this is...
There's an anemone too.
Yeah.
I'm glad we got a specimen of it a few cruises ago, and then of course that black coral with possibly the same thing, I'm not sure, earlier today. Because I think this could be a brand new species. Maybe even something more than that. These tubes at the top are just unique. Right there.
Weird.
Great, thank you, pilot. Appreciate that.
Okay.
Right, video's clear.
Okay. Go
Chris,
back.
I don't know if you noticed the sponges. We were coming in just to the right. I'm not looking at the screen right now, but it had some long branches, skinny branches, and
Oh, they want to look at that.
Okay, yep, go ahead.
Yeah. Pilot's got you there. He heard you. And we're looking right now, Scott.
It's a partial.
So it is a dying Malteria where their
Yeah, co-pilot, go ahead. Take
They're-
that, please.
Tilting.
side projections have survived, but not much else on the sponge.
Could we rotate down
Yeah.
a little bit?
Now, has that got a lot of stuff on it or what, Chris?
That is just like what happens when
Lose the lasers.
you
When commensals go bad.
yeah, when you've just left it like if you
Is it all right to zoom more?
That's a lot of
you've got an
associates.
abandoned house and everybody moves in. Yeah.
That's a good analogy. So we've got squat lobsters, zoanthids, hydrozoans. We got everybody, and that's actually this tall white stalk to the left is the original, the remaining live part of the original sponge, which is all yellow right now, in between all of this mess all over it,
Yeah.
and no stars.
Nope. No worries.
Okay.
I'm sure something will come up.
We're good when video is
Okay.
good, pilot. Thank you.
All right.
You guys ready for a move? Or not quite yet? Maybe.
All right, video's good.
Okay. Copy that.
Maybe just go look and see what we got out there, and then I'll call it in, probably.
We're full wide.
All right, thank you. Yeah, and we're about ready for a swap, too. So
yeah.
we want to wait for that.
Pilot, the
Get
side
out of here
branches
so they're
survive
not
and nothing
caught
else on that
behind.
sponge.
Right.
I'll pull up.
They've
a
got
Okay.
tiny little
Sounds
spines
good.
sticking out of the side branches, and maybe that's enough defense, or I don't know.
Okay. Let me get up a little bit.
Well, I mean, I guess it depends on how holistic the body
Pushing it.
is. I mean, you know, if
Looking
the
70
central part dies,
degrees.
it might mean that the peripheral
Heading
bits don't need to die right
300.
away. They can persist on their own because there's not as much to persist on. You know, they can just
Come
filter
up and give
feed
you a
or
little room here.
survive for a
There's a
while
fish.
longer.
Okay.
Right.
Where do you see it? Oh,
He's
Is that
a
fish?
fast guy.
Oh,
Where was it?
some kind of tiny little
Zoomed right on through, yeah.
Yeah.
Well, things are
Yeah, it looked
definitely.
like a fish, maybe a midwater fish, like maybe cyclophonia or something, but it was hard to tell.
Ah,
Sorry, Bruce. I didn't even see it. It must have gone by so fast.
see, this is like prime real estate.
Is that a
for all
starfish
sorts
up there at the top, or is that
Where
the
is it?
base?
Oh, could. Well,
Top.
yeah, look at the white thing
It's
up
on
there.
top of a rock.
Could be.
Yeah,
Could
let's-
be.
Yeah, if you wander over there.
Yeah.
Could we get lasers on?
Yeah.
Lasers coming on.
There's hope here, I think, Chris.
I hope
Thank
so.
you.
Looks like
Looks
legs.
suspiciously like a base, though.
I think it looks suspiciously like a
starfish. Me too.
Oh,
That's the wrong way to turn that handle.
it's nice to be.
Port side is
Stand
first
by.
try.
Yeah.
Moving in.
It is a starfish, isn't it?
Is it?
Sure it is.
Sure, it's not just a broken branch.
No.
No, it's a
Yeah,
starfish.
it's definitely a
Oh,
starfish.
Don, you're the hero of the day.
Yeah,
You
way
get
to
a-
go, Don.
You get a big star.
Yeah. So,
Finally.
Finally.
so I think
Finally, I did something good today.
What are you talking about? You're always doing stuff good. Many people who are listening may not realize, but Don was the pilot over the Johnson Sea Link, which is a submersible that is operated down in Florida, and that was the first sub that I was ever on. So this is kind of a nice revisitation. Let's see what it is.
Okay, let me come to a stop here. Oops.
I got an idea, but I'm not going to throw it out
That's
until...
too close.
Yeah,
I
Can't
think I
tilt
know
down.
what it is too, but
Yeah.
let's take a quick
Current
look.
was
just
going
in
to take
case.
me back a little bit there. Okay, you want to go for a partial zoom on that?
I got your tilt.
Okay, thank you.
So that's a partial.
I'm also curious about that track that
Tilting
was
up.
to the west of the sea star. There often is an animal at the end of a track like that.
Yeah, you got too much vert on.
Okay, yep. I want to get a different position on this. And
Starfish
come
was
around.
announcement
Or
bomb
wide.
this
Yes.
time.
Yeah, all right, thank you.
It's getting to be pretty
Let me get
regular.
over here to the
bit.
right. And
So what do we...
yeah, I'm
Yeah, I
repositioning.
think,
I want to get looked down on it a
It's
little bit more.
pretty clear that that's Henricia.
Okay.
Henricia is a very problematic genus of sea stars. They occur all over the place in cold water, from the Arctic to the deep sea to the Antarctic. They're, as far as I know, primarily sponge feeders. They're very plain-looking in appearance, which has made identification of species throughout the world very problematic as far as figuring out where one species begins and where another species ends. There are lots of also very interesting evolutionary problems because you are trying to look at
Try a
how
little
these
joy
have
lock when you get things
sort
balanced
of
out.
diversified in
If I can
certain
reach
areas.
Use your thumb.
And you know, there are places like in the Aleutians where there's a ton of these.
You still have
and
yaw
they're all a little different from each
Enjoy
other,
lock.
but there's a complex of them, and trying to figure them out has been very difficult. But they're ecologically important. They occur, prominently in several ecosystems. Some of them are brooders. They can actually have little baby sea
not
stars.
working.
Actually, as much as I'm appreciative, if it's we can sort of look at this trace on the left too, if that's a little easier.
He's trying to set down.
I'm
so
trying to
Josh
set
can
down,
take,
so
take
yeah,
over.
yeah,
Oh, all right. No, sorry. I'm not trying to just sort of
Yeah,
dismiss it.
no.
If you can set down, that'd be
Yeah.
great. But if it's a big
I'll
pain,
just.
then
Well, no, I mean, he's trying to set down because there's a watch change going on here.
oh well then. Oh yeah, no, okay. I should have been
There we
more
go.
attentive. Thank you, Don.
All right.
Thank
Yeah,
you for
that was Don's exiting find right there. So that was a pretty good way to end his shift here.
I commend you, Don. Thank you.
So it's a pretty scene while we're waiting for the change in the front row. We are at 2,010 meters. We're on Wetmore Seamount, or what may be named Wetmore Seamount in the future. We're allowing the next shift to get settled in. So they're going to sit here a moment while the navigator on the previous watch is updating the new navigator coming in.
So Josh, we never actually got a
Eli,
zoom
or
on
excuse me,
this.
Levi is leaving,
Yep,
and
come on
then
in.
Sean is going to be taken over.
Give your tilt back, Josh.
Yeah, thank you.
You got it.
And in fact, if there's a way to get a zoom on that brittle star onto the right, that would be great as well.
All right, come on in, video.
That's full zoom.
Look at those little ball-shaped sponges there,
Yeah.
demasponges.
Oh wow,
on
look
there.
at them. It's
Yeah.
like they're forming. Oh yeah, see that's what I bet they're feeding on.
Yeah.
The Henricia. I mean,
Yeah, come in tight.
That's full zoom.
there's also some people have suggested that they're filter feeders, but I'm far more inclined to accept the fact that they're just predators that they feed on just very, very non-moving things.
So Josh is now the new pilot, if I'm not mistaken.
That is correct.
Before we do intros, there's one thing behind the Sea Star after we're done with this guy that it's more of confirmation. There's some question as to whether there's Metallogorgia here so far, and there's one of these a little bit, keep going a little bit beyond it, right there, and that's what I was looking at, perfect. And that's not Metallogorgia, it's another stocked Chrysogorgia, and it's got a squat lobster in it. So I think we've been seeing a lot of those, and not
Metallogorgia.
Not Metallogorgia. So thank you very much. Appreciate it.
Some nice shots of, I think, Ophioplinthaka and Ophiacanthid brittle star living on this, what is it, Primnoid?
Yeah. Yeah, I was just typing that in. So this looks like Calyptrophora, maybe even Calyptrophora angularis, which is the host coral for these brittle stars. That's in the family Primnoidae.
Hey, if you're good science.
Yeah, we are. Thanks.
Come on out.
Appreciate
All
it.
right.
So,
what else would you like to see here? There's a trail you want to look at.
yeah, it was just right above the lasers.
Yeah,
I
come on
think that's what he was interested in.
It's not quite full.
Is that one of your sea cucumber trails, or
I'm not sure. It could just be a sediment-filled
crack or something?
crack or something. Yeah, that's okay. I think. Thank you. We can move on.
All right, come on out.
Yeah, I don't see anything immediately evident creating it.
All right, anything else here?
I
It
mean,
doesn't look like what I thought it might have been. I thought it might have been one of the trails from a monoplacophora or one of the gastropods, but it's
Keep
not.
up that
So
thinking, Bruce.
Thanks for zooming on that.
Oh no, thank you. Yeah, no, we can move on. I think
Yeah, copy that.
this was the first Sea Star today, so if you see one, buy a cookie at dinner or something.
All right, copy
So-
that.
Josh, who do we have in the front row now?
So, piloting D2, this is Joshua Carlson. To my left is Sean Kennison for navigator. To my right is Levi Unova on co-pilot. On shading, video chair is Annie. And clipping, we have Bob Nutt, and we're all engineers and videographers for the Global Foundation for Ocean Exploration.
All right, thank you very much. So just updating you, Josh, we've seen a lot of the stuff. We're trying to find new and different things.
Yeah,
Seeing
copy
all
that.
these big the whip-like corals and small chrysogorges and bamboo fans. But
Yep, so the direction Sirius is facing,
And then
300.
I think we were
That's
trying
the
to
direction we
sidle.
wanna go.
You're over on the
Sounds good.
north side of this
Yeah,
ridge
my
too,
sonar
and
looks
it was
good.
more productive. Oh, look at this large Anthomastus. This is new.
Let's get this guy. Come on
Yeah.
in, video.
It's a partial.
Okay,
Let me
so
get stable here. All right, go ahead in.
and this is a different type of mushroom coral than we saw earlier. This is Anthomastus tahinotus is, I think, what it's called. And it's a beautiful, these are probably one of the largest Anthomastus species that we have out here in the Central Pacific. It's getting very, very large with a very thick stalk. Maybe not as large as the West Coast. off of California and that area, but these are pretty good sized out of here.
It's coming tight.
That's full swing.
Nice.
They're all beautiful animals. Really enjoy looking down the feeding tentacles of these.
Yeah. I think we're good, Josh, and Annie's good.
All right, let me know when you're good, Annie, or if you want another shot.
If we can just get a steady shot of this for just a minute. A second.
Oh, that's beautiful. And we're looking at the reason why this is an octocoral. There's 8 tentacles around each of those polyps, and they're all pinnate, which means they have little pinnules along that make them look fern-like. And those are pretty distinctive of the octocoral group as opposed to the hexacoral group of cnidarians.
Come out a little bit.
Black corals are hexacorals, as are zoanthids and anemones and hard corals and sclerotinia.
Full wide.
They don't have that type of tentacle arrangement.
Is the bridge?
Hey, you ready for shipment?
We are.
30 meters at 300 degrees at 0.2 knots.
Pilot, I wonder if you
That
could
is
take a quick swing
range
left.
30,
just
bearing
to bring the
300,
camera.
speed
I thought I
0
saw
decimal
something
2.
yellow. Yeah, that yellow. It may be overgrown,
Here we go.
but it also could be a plexaurid.
This guy center on the lasers?
Yeah, that's it.
All right, come on in video.
Oh, it's a paragorgia that's overgrown by zoanthids, the paragorgia coralloides or something. Did we record any of these? I don't think we have
today. Yeah. I mean, we've seen lots of coral colonies overgrown by
ROV nav,
zoanthids, possibly even more than one species of zoanthid, but I think this is the first time that we've seen paragorgia overgrown by a zoanthid.
Do you have any more zoom, or is that max?
No, I got a lot more.
Yeah, go ahead.
Okay.
So, you know, a lot of these paragorgias that are overgrown, they're overgrown all except for their little growing tips. And I don't know, you know, the zoanthid can expand its colony if the coral can expand its colony. You know what I mean? I wonder if there's something going on where the zoanthids don't go all the way to the tip and the paragorgia keeps growing, allowing, and as soon as it's gone far enough, another zoanthid polyp can then go further. You know what I mean? Does that make sense?
So what you're saying is the paragorgia has to have, since it's the intrinsic body, it has to create more body for the zoanthid to command?
Right, and so it can do that maybe because of the tips are at least still growing and haven't been over.
Interesting, yeah.
I don't know.
Interesting. I'm not even sure how... I'm not even sure necessarily how this works. I know that you can look at intertidal sea anemones and see that they're constantly fighting for space, you know, with their stinging cells pushing back and forth. But we've mentioned how these sort of commandeer the bodies of these paragorgia, but I don't know that I've ever seen exactly how. It's just with the, I guess what's with the mats or something that they colonize with, the sort of tissue that they use.
Yeah, I guess so. The
For a foot wide.
the mesenchyme, what do they call that tissue?
Anyway, I know what you're
something
talking
like
about.
that, right? Okay. So we're in search of more unique critters. So got a stock sponge off to the left, and a lot of these whip corals all over the place.
Just do a snap over here. Come on in.
Yeah.
Okay. How's
So
that?
go ahead and type. I just want to see what this is.
Oh, okay.
Yeah, and
Just the
could you zoom up on the stock coral as you're pulling out?
Come on out.
Yeah, right there.
Which guy?
That one.
Stock sponge.
Stock sponge?
Yeah.
Oh, okay. You said coral.
And there's
so I was
something
confused.
purple, Chris, right beyond, excuse me, Josh,
like a
right to the
hologram.
left of the ball on the stop sponge.
Yeah, it looks like a sea cucumber.
All right, let's get
Is
the
that what it
sponge.
is?
Yeah,
Come on in.
I can see the gut from here.
Yeah.
And it looks like the sponge has a crinoid on it.
All right. So. Okay, so I know this is Caliphacus. The problem is it's got that double branch with a different ball for a double branching Caliphacus sponge. So
Very odd looking thing, isn't it?
yeah, so Caliphacus is a glass sponge. It's got spicules or microscopic parts made of silica. And this is in the family Rosellidae. And they include vase-shaped sponges, that family, as well as these beautiful stock sponges. And they're difficult to untangle sometimes what their taxonomy is. So thank you, Pilot. That'll work.
Okay, let's get the sea cucumber real quick.
Yep. You want me to go in more?
Yeah, go ahead.
Okay.
Yeah,
Let's full zoom.
it's another nice sort of translucent bodied sea cucumber. You can sort of see that dark array of feeding arms attached to the bottom, probably where it's feeding. There might even be, I think there's a little. Are we at max zoom?
Yeah, we are.
Yeah, I can get closer.
Yeah, I'm just trying to see what that little orange and white thing on the surface might be. But these are essentially detritivores. They like to roam around on the bottom of the seafloor and eat all of the ooze that is present amongst the sediment, and so they digest all of that together. And what you're seeing in the gut is all of that sediment in the having just been swallowed and eaten by the animal, and then
move complete.
Eventually, it sort of works its way through the intestine and gets pooped out as a sort of clean sediment, which is actually very important for the ecosystem because it processes microbial fauna as well as clears out the organic stuff. This is also kind of cool because I believe that what we're seeing is a eulimid snail.
Oh, we're at full zoom.
So this is a little parasitic snail. You see this little triangular bit that's on the tip there? Yeah, it's a parasitic snail. And so what they do is they have a proboscis, and they just sort of like a big, like a juice box. They just kind of go dunk, and they put their proboscis in there, and they suck the juices out of the host. And these are found in all sorts of echinoderms, from sea cucumbers to crinoids to sea stars. And this is kind of nice because I don't think we've ever seen one in situ before. The sea cucumber may or may not mind. It's hard to say. But now we're seeing kind of the feeding surface sort of blowing up a little, so we can see that. But it's kind of neat, kind of nice to see this sort of ecological interaction in its habitat. So good to know. Thank you.
Yeah, come on up.
Right. We're full wide.
And I'm going to center up on you again, Levi.
Okay.
Sounds great.
Glad you called that.
Video's
Chris.
gonna switch out.
Right.
That eulimid was worth it.
Oh,
Thanks,
sorry.
Chris.
Yeah. Sorry, I was.
No worries.
Had my microphone off.
It is heartening to see that I'm getting the hang of it.
You know, there's a lot of switches in front of us. So sometimes we can push the wrong one. One of these, I guess it's a J-clade bamboo. There's another one of those sponges, the turbocharger sponges. So, you know, we're... Oh, and there's a shrimp that just came through. And that's an Acanthophyra. And why is it an
that what it's
Acanthophyra?
called? Acanthophyra.
Go ahead, Ed.
Yeah, I answered it eventually. And what's the common name? My common name for it?
What's the common name for that?
like
A drunken shrimp, but that's my common name. That's not a real common name. That guy cannot swim straight. So I think a Canthophyra is a sort of the shrimp version of a Roomba vacuum cleaner. So it just looks like it's got this random path, and it's bound to intercept stuff and get the dust and dirt like that shrimp is by just wandering all around in all different directions. Wow, the tracking has sort of gone a little bit haywire, hasn't it?
Oh dear.
Another Caulophacus sponge. And another sea cucumber. We could pass that one by, but something's... Oh, it's a brittle star baling in it off of the dead sponge stock.
Crinoids?
Yeah, pair of crinoids. Is that a sea star? Or
Yeah.
what is that?
Come on in.
Oh, yeah, brittle star, maybe.
Yeah,
Brittle star.
Brittle star, okay. Oh, maybe that's the guy that just bailed off of it. Yeah. It's a funny behavior of them jumping off of whatever they're perched.
Yeah. Oh wait, what's that? Just on the lower screen, there was. It looked like a sea urchin with spines coming off of it.
Where?
There.
Oh, that.
Oh yeah.
Oh wait, is it a sea urchin or a forearm?
Or a demosponge, starburst demosponge.
Oh.
I think it's on a little stalk.
Interesting. Yeah, zoom as is convenient, please. Thank you.
All right, go ahead in.
That's a demosponge.
Are you sure?
No, I'm not sure now. Is that an
wow. Well,
yeah. It's an urchin.
Is it an urchin?
Yeah, it's an urchin.
Huh. That does
pretty
look like
That's Maxim.
flattened urchin.
Thank you.
it doesn't leave a, I mean, I'm not even sure. I mean, it kind of looks like a sideroid, but not at this depth. And it's, how big is that?
That is teeny.
Come out and get the lasers. It's very, very small.
Oh, nuts. Yeah, that's too tiny. Okay.
Yeah, come back in.
Interesting. No, it's a fascinating urchin. I don't know. A pedinid, maybe? It has kind of that spiny pedinid appearance.
Sort of.
Sort of. Yeah. Yeah. No.
That completely barren top is pretty strange.
Is it upside down?
No, and it's not upside down, is it?
No. I don't think so. Because if it was upside down, there'd be a bunch of stuff around the mouth.
Yeah. Okay.
That's just a bizarre. Huh. So frustrating. Okay. There's only so much I think we can. That's fascinating, but I don't.
Come on up, video.
It's too bad it's not on a rock or something.
Yeah. Chris, let's leave out hope that was a baby one of those.
Permission to put another move?
Yeah,
right. You never know.
I'm ready
so
for
here's
it.
one of the mysteries of
I'll
deep
put
sea
it a few
exploration.
meters at 300.
Is you come across a small sea urchin like that, and
Sounds
you
good.
don't know if it's a juvenile of something that's known that is an adult,
6
or if
years
that's just
tracking
the
looks
adult.
pretty good.
And because there are
Sirius
lots of
came back.
species, adult
D2 is
species,
still
that are simply very, very small, and there's a reason. There's usually some adaptive reason for it. It can.
Bridge, go ahead.
And so usually this is why.
Yes, sir.
Studying deep sea animals can be such a challenge because you don't really have the full context.
Range
and
30 meters, bearing
there are lots of
30
animals that
degrees,
you don't have a really good diversity of
Coming up.
individuals to sample. You don't know what's normal for any individual species. So something like that is a sort of a frustrating mystery, and especially if they're described as a new species, sort of incorrectly. It turns out, and if it turns out to be another species, it's often difficult to tell what's going on.
Quick to see what that is. That's a base.
So,
Alright, coming up.
I just noticed John Smith's comment about the drunken shrimp, the Acanthophyra, in the event log. He suggested calling it,
on that
since we're calling
right
it the drunken shrimp, he suggested calling it a shrimp cocktail.
Oh!
Coming in.
Good one, John.
It's a Munidopsis, the white squat lobster behind the dead sponge.
Have we seen one of those yet?
Wow, there's a lot of stuff on that. Little snails, squat lobsters, hydroids. That's neat.
It's a dead Walteria sponge. Walterias are some of the kind of stranger vase sponges in the family Euplectilidae. It's a glass sponge, but they have this very weird morphology of this lattice work structure, and then all of these lateralia is, I think, what they're called. These spines.
They seem to sort of persist longer than the animal itself does.
Yeah,
interesting.
I don't quite understand that. Yeah,
These are the kinds of things
these are anemones too on here because they're individual, right? They're not like zoanthids.
I'm not sure I can tell from here, but yeah, they look
See,
individual.
they're not connected.
Yeah. Huh. It's a treasure trove for sort of discovering different little new species.
Okay, we're fine when video is. Thank you.
Coming up.
So we're continuing upslope. We're slowly coming out of the little bit of steeper area, and then we're going to go into a little bit of a flatter area before we get to waypoint 2 here. So we started flat and then went into a steep area. Now we're going to have a flatter area.
A little amphipod.
Trying to think of what they look. There's something funny about the way they swim. I don't know. Does that guy have any hope of swimming against the current? I don't know.
Huh. Is it a Hyperiod? Huh.
I don't know what that is.
Yeah.
What's your mic there, Levi?
Sorry.
What's our off bottom time?
1 hour from now, I think.
Yeah, 1:23 UTC. So yeah, just under an hour.
Okay.
Pilot, I didn't hear him. What's our off bottom time?
Off bottom time is going to be 1:23 UTC, so just under an hour.
Just under an hour. Okay, thank you. More of the various type of unbranched corals and we've been seeing today. as well as some of these fan shapes. Another corallium to the right.
Is it just me, or did the Walteria suddenly start dying?
Well, here's one right here. Well, yeah, there's a whole cluster of dead ones. But
I mean, that could just be sort of a wave of recruitment.
Come in on this guy.
yeah, look at the Calyptrophora. That's got a lot of brittle stars on them.
Yeah.
This is Calyptrophora angularis, I think.
Sort of crinoid, okay. See, why can't we see one of those